Hello, and a very warm welcome to everyone joining us for this exciting Select Science webinar. My name is Cameron Smith-Craig. I'll be conducting today's session, and I'm delighted to be joined by our very special guest, Sarah Stahl-Rommel. Sarah is the research team lead for microbial monitoring and technology development at the NASA Johnson Space Center in Houston, Texas. Toward that goal, she has driven the development of a complete end-to-end sample-to-sequencer process for microbiome assessments and oversaw its successful implementation during the NASA Extreme Environment Mission Operations, or NEMO missions 21 and 22, which took place on the ocean floor. As the project scientist for the Genes in Space 3 flight investigation, Sarah has since worked to successfully transition the NEMO method to the International Space Station, which resulted in the first ever identification of microbes collected and cultured off Earth. Today, we are fortunate enough to hear from Sarah how her team of fellow NASA scientists were able to achieve this goal of bringing real-time microbial monitoring to the International Space Station, with a sneak peek of NASA's plans for the future as missions such as Artemis 2 take us back to the moon and beyond. Throughout the webinar, please feel free to submit any questions you may have. To do this, simply hover over the purple speech bubble icon to the left of your screen and click to open the submission tool. During the presentation, we will also be circulating some poll questions. If you could please take a moment to submit an answer when prompted, it would be greatly appreciated. Thank you. So without further delay, I'm delighted to hand over to Sarah and would like to thank her for presenting to us today.
All right, thank you so much for the introduction. I'm really excited to be here today to get to talk to you all about the work that my group has been doing on developing spaceflight microbiology and really moving it forward beyond the culture methods into some molecular technologies. So excited to share all of that with you. As you might be familiar with the International Space Station, that's where we have crew members living up and orbiting the Earth. And that's where the majority of all of NASA's focus is in this low Earth orbit environment. And we get to do a lot of really exciting research there. But before I get into that, I wanted to talk about kind of the future goals of NASA. So the International Space Station is planned to be going away in 2030 and moving more towards the commercial aspect. So NASA is really interested in going towards these deep space missions. So we're focusing on returning to the moon through the Artemis program and with the Gateway habitat that you can see in the lower right side of the screen. This Gateway habitat is going to be like the new ISS. And by using this, we'll be able to orbit the moon and allow the crew members to explore new areas of the moon that we haven't seen before. And it will also allow us to use this as a launching pad to move on to Mars and other deep space exploration missions. So we can use the Gateway habitat to assemble this Mars vehicle to move forward. And there's a whole bunch of challenges that come with moving towards these deep space missions. Like with the International Space Station, the crew members are in constant communication with the ground, with mission control. They can reach all the experts. They're able to receive resupply missions and all things like that. But when we move to deep space missions, there's a lot less accessibility to the Earth. So the crew members need to have increased autonomy and be able to do things more on their own. When we're on the moon, they won't be able to return to the Earth. It'll take them a few days. And once we go out to Mars, it takes months to years. So it's really important for us to take advantage of the International Space Station while we have it to keep expanding our knowledge of spaceflight and microgravity so that we can apply it to these deep space future missions. But the majority of my talk today is going to be talking about all of our work that we've been able to do on the International Space Station. So currently there's 7 crew members orbiting the Earth right now, and the ISS has been constantly inhabited for the past 22 years. So you can think about how there's a lot of really fascinating research that has gone on and what is microbially happening on the International Space Station. So when you think about NASA, you might not think about microbiology, but it is a really important aspect of spaceflight. So as microbiologists at NASA, we are in charge of mitigating any kind of microbial risks to the crew health, the crew safety, and their performance during their exploration mission. Part of this is making sure there's no adverse effects of the microbes on the vehicle itself. We also have a big area of emphasis on research. How does the microgravity environment impact the microbes? There's a lot of research showing that some microorganisms actually become more virulent in spaceflight, which is bad because you don't want to make it more harmful for the crew to get sick. Another big area that we focus on a lot, and I'm going to talk a lot about in my talk, is the new technology that we're looking into to expand our ability to assess microorganisms. So spaceflight microbiology, a big part of this is applying the requirements for microbial acceptability limits. And we look at those specifically in air, water, and surfaces, and foods. And by these acceptability limits, they're based off of how much is there and what is there. And by saying what is there, is it medically significant? Will it have any impact on the crew? So our acceptability limits are listed at the top. And as part of doing this monitoring, we do routine pre-flight and in-flight monitoring to check out to make sure that they're within these limits. That involves either monitoring the launch vehicle, anything that gets loaded into the vehicle in, like, say, the cargo bags, and then the food. Even if it comes from a grocery store, it will get repackaged, and then we have to test it in our lab to make sure that they're within our microbial standards. A part of that is if they are beyond our standards, we implement a remediation routine. And then on top of that, taking everything into consideration, we're responsible for delivering this certification of flight readiness status. So when you think of all of those spaceflight movies like Apollo 13, you see them going around in mission control and they're saying, go, no go, go, no go, and saying, do I approve that this mission can happen or not? Well, in real life, there is an actual go, no go for microbiology requirements. It's maybe not as glamorous as it is in the movies. We're not in mission control. We're just in a conference room, but it's very exciting to know that microbiology has that big of an impact on a mission to say, yes, it can or cannot go. The part of the in-flight monitoring that the crew members do is for air, surfaces, and water. And so we do this on a quarterly basis. And the crew members, for surfaces, they can collect a swab or use the contact slide that contains some agar media. We use two slides. We use a bacterial slide for TSA and a fungal slide, a sabdextrose with some chloramphenicol. And they can touch that to a surface. And they also do the same thing with the agar media for air using the microbial air sampler. And they'll let those grow at room temperature for 5 days for bacteria, up to 7 days for fungus. And then for water, they use several filtering steps where they get water from the potable water dispenser, and they can filter it onto this microbial capture device here. And on the back side, they add a little bit of media, and then there's some dye in here to actually produce these little blue dots, and that allows the crew members to visually see the growth that occurs on these plates. Additionally, we do a coliform sampling. We can take water from the potable water dispenser, put it into these coliform media, and let that grow for 2 days. So all of this is done, like I said, on a quarterly basis, and it requires the crew members to collect the samples and then culture them on the space station. So by doing this, they're taking microbes that are on the surfaces and growing them up, and visually they'll look at the plates, and that's the information that we get back on the ground is there a lot of growth here, or is there a little bit of growth here? They match it up to a card to say A, B, C, or D, is there a lot or a little? And that's the only information that we get in flight. Those plates have to return to the ground into our laboratory so that we can identify what are the organisms and then complete our microbial risk assessment of what is there. The only information that we can get on board right now with this method is the call forms, and that will tell you, is there a potential of a fecal contaminant in the water system? And that would, a yellow bag like this picture is negative, a pink bag would be a positive. And that's the only identification-based information that we get during flight. So I really want to stress that we have to return all of this stuff to the ground to figure out what it is, and that can take up to 30 days because they have to sample it, let it grow on board, then it has to get loaded to a vehicle, returned to the ground into our laboratory, then go through our standard laboratory methods. Luckily, a lot of the 22 years of data that we've shown from the ISS culture data is that we know the ISS is a pretty clean environment. Most of our fungal counts are not detectable. We don't get a lot of fungal growth. This figure here on the right side shows what the ISS detection limit is versus the bars are showing an average growth of bacteria and fungus in your home. So you can see that most homes are above the ISS limits or right around it. So we know that the ISS is pretty low microbial loads. The crew members do cleaning activities every week. So we work really hard to keep it clean. There's also a lot of engineering controls in place with our HEPA filters and stuff like that. The main microorganisms that we've found over the years are human-associated, like skin bacteria, like Staphylococcus, Micrococcus bacillus. We also see Aspergillus and Penicilliums more often. So even though we know that the ISS is a pretty low biomass environment, there are always these occasions where we get pictures from the crew members and they say, hey, what is this? Should we be worried about it? Is it microbial? Is it alive? Is it just a stain? Because all the materials that are up there, there are some of these like soft fabrics and stuff like that, that sometimes, like with this picture in the corner over here, it's showing a panel where they would strap wet towels to it to dry. And so you could see that some of the moisture then got into this panel and it caused, this case, an actual fungal growth to occur. But in order to figure that out, we had to get samples collected by the crew members and sent back down to the ground. So our goal here is saying, how can we make this more autonomous for the crew members to do some of these investigations themselves? And instead of bringing samples back to the lab, what if we could bring the lab to the samples? So technology over the years, I like to show that we've progressed in our spaceflight vehicles and our missions and our objectives, but the microbiology has stayed the same. We've been using these same culture-based methods since the beginning of human spaceflight. And what are we going to do now that we're moving towards these deep space missions where we're not able to return samples to the ground? We need to really progress our technology to keep up with that. So 2016 is what I like to call the dawn of the molecular space age. So in April 2016 was when the first molecular biology experiment was completed on the International Space Station. This was part of Genes in Space program, which, if you're not familiar with it, it is a fantastic student program where students from 7th to 12th grade can submit proposals and win this competition. So if you're interested in, I'd highly look into that more. And then in August 2016 was when my experiment was completed on the space station, and that was when we did the first DNA sequencing in spaceflight, so showing that this MinION sequencer could be used in flight. Before we get into all of those details, I just wanted to highlight again Why is sequencing so important in space? So I think I've made a very good point about why we care about it from a microbiology standpoint, because we want to be able to diagnose things in flight and reduce down mass. But there's a whole nother area that I want to highlight, is that there's many other researchers doing work on the International Space Station, whether it's human associated work, there's a bunch of animal, we have rodents up on board, we also have plant habitats. There's endless science and research that's constantly going on at the space station that could benefit from this technology. Also thinking about the use into medical operations in the future and how we could use this for diagnostics and biomarker tracking and countermeasures. And finally, this is really fascinating to think, how can we use this technology for life detection on other planets? We have a colleague, Dr. Erin Burton, who started the sequencing project with us, who's really into the astrobiology and looking for life and showing that we can use this technology to sequence unknown nucleic acids. And so it's very exciting to see how this can grow and be used. So our first sequencing in space, I mentioned, was in 2016. This was with the biomolecule sequencer payload. And we flew the MinION sequencer from Oxford Nanopore Technologies. And if you're not familiar with it, it is a really small, portable, handheld sequencer that uses these flow cells. And the flow cells have about 2,024 little nanopore proteins in this chamber here. And this video is showing that the DNA is traveling through the nanopore protein. And as it goes through the hole, it blocks the current, and that is what can get decoded into the basis of the DNA that we're seeing. So by using this third generation sequencing technology, we can take it to space, or there's many researchers that are taking it out into the field. Because without this, we wouldn't be able to do this, because we know that the Sanger sequencing that we use in our lab, or the Illumina, those require large cameras that are really sensitive, and they would not be possible to use only from the fact that they are sensitive, but also they're large and heavy. And I can get into some more of the spaceflight requirements in a little bit. But with this study, I prepared samples on the ground of a mixture of mouse, lambda, and E. coli DNA, and we froze them and launched them to space. And then astronaut Kate Rubins thawed them and loaded them into the sequencer in flight, and then I thawed them and loaded them in the lab, and we compared the results, and everything matched up perfectly. So this was a huge, paradigm shifting experiment showing that we can actually use sequencing technology in spaceflight. But what's the point of having this sequencing technology if we don't have a way to actually prepare samples? We don't want to keep making samples on the ground and then launching them to space. So that's led me to this goal with my colleagues to develop this sample preparation method. And so our main focus was starting with this swab to sequencer, with a goal of comparing it back to our culture-based methods for surfaces. So the crew members were collecting swabs and going to culture. So we were trying to figure out, how could we collect swabs and go to sequencing to get the same answers, and how do they compare? So to develop this method, focusing on how do you make a spaceflight compatible sample preparation method. So the first thing I did was go into the lab and think about, what do I use for my low biomass sample preparations? And immediately I realized I couldn't use any of these kits or these large equipments because they are heavy, or they have toxic chemicals in there that couldn't be within the spaceflight requirements. This picture shows the MWA, the multi-purpose workstation, where the crew members get to do our science. And you can see it's basically just a little fold-out table. There's not a whole lot of space. They're back to back with another crew member that could be at that table, and there's a lot of other activities going on around them. So it's a very limited little lab bench space area. They don't have access to these large laboratories like we do on the ground. So those are some of the key things that we had to take into account when developing a sample prep method. Was the number one thing is the crew safety. So we really want to focus in on the toxicology of the reagents that we're sending up there. From both a flammability standpoint, we're not allowed to send any ethanols or risks of flammability. Also thinking about the off-gassing of things that you're sending up there and how that could impact other things in spaceflight. We have the capability to send things at a higher toxicology level, but you would have to keep it contained within like a glove bag or a glove box and add additional layers of containment based on what you need. And we had a big goal of being able to maintain open cabin operations. So being able to just work on that MWA station, because once you have to get into other boxes and it just makes it clunkier and harder to schedule with crew time, which is another large area that we needed to iron out for the sample prep. Is the crew are very busy. They have a lot of different activities going on. So you really want to make sure that it's streamlined as possible. Also, the storage conditions are limited. We only have ambient temperature for degrees Celsius and minus 90 degrees Celsius. So finding chemicals that can fit into those conditions and having a shelf life that's long enough to not need constant resupplies. Because like I said, we get missions resupplying items to the crew members, but it's not super often, and it's expensive to send stuff up there. So again, just finding things that can be flexible. And then from a science standpoint, we had to pick out a lot of interest in finding how do we target these really low biomass samples that we know are existing. So looking at, we looked at several different swab types. So focusing on, we looked at some foam swabs, some nylon flocked swabs, some polyester swabs. We ended up with a alpha knit polyester swab in the end, but finding what is the recovery that we're getting off of these swabs, and then leading into that is what is the most optimal DNA extraction method. There were several different things that we tested through this, because when you think about in the laboratory, you would traditionally use like a large bead beating machine. So we looked at using a handheld bead beater. We also looked at using some heat and enzymes, and I'll talk more about those throughout my progression of our work, but ultimately we found that processing the swab head in the heat and the enzymes was really the best way to do that, to get the best extraction. And then finally, purifications. We couldn't just use a spin column like you would use in a lab, so we were able to switch it out for magnetic bead-based purifications, and those have been working out really well. So once I had a preliminary method for sample preparation, we had this opportunity to participate in NASA's Extreme Environment Mission Operations Analog, where the crew members are living in this habitat that's on the bottom of the ocean. It's about 60 feet below sea level, and it's off the coast of Key Largo in Florida. And so this is a good analog for how the crew are on the International Space Station. They're isolated, they're in the stressful environment, they have limited space, and they're busy with all sorts of activities. So NEMO 21, this is where I feel like my sample preparation that I did in the lab, I had an idea that we had worked through of like, this should work. But going and actually applying it in the field had a really big impact on how do we actually do sample preparation in an operational setting. So we had 6 crew members of various backgrounds. Some of them are scientists, some of them are engineers or Army, Navy pilots. Like, it was a very diverse group to being applying it to. And we learned a lot about the importance of your flow of your operations. So how do you package everything up? In a laboratory, you don't think about that. You just go and you grab your kit and you follow your instructions. But with this one, we had to have everything individually packaged into like a one-time use format. How do we have your crew procedures that are laid out in a easy to follow manner? We had opportunities to do some crew training with the crew, but how do you iron out like what are the important things to hit? Because there's obviously you have to review how do you use a pipette, how do you load the sequencer, how do you collect a swab. Um, so this was really eye-opening to just think about doing biology, molecular biology in an analog environment. So we learned a lot from NEMO 21. While we were doing this, this was our preliminary protocol. So we were using a larger swab, you can see in these pictures, and the bead beating. But those were taking a lot of time, and our extraction yields weren't like we wanted. So we had to go back and redesign our workflow. So we picked a new swab that was smaller. We picked a new DNA extraction method. So we switched over to this quick extract from Lucigen, and we're able to take advantage of processing the swab in the extraction buffer and heating it, and then updating our crew procedures. So streamlining everything. And the data that we got from this was so cool to see. We swabbed like a phone, a shower, and a table, and you could see the mixture of human-associated microorganisms and ocean-associated microorganisms. So seeing that there were things being found in the environment from the habitat. So this was really a big milestone for us, getting comfortable with operational sample preparation and deploying a method. So from our experience at NEMO, we were able to start transitioning some of this sample preparation to the International Space Station. And in 2017, with the Gene Space 3 payload, we did the first library preparation completed off Earth. So we got to work with astronaut Peggy Whitson, and she did the first pipetting, open cabin operations. There was so much unknown about this. Like, we didn't know if a normal Eppendorf pipette would work in spaceflight, or if you open the tube, would the liquid stay in the tube or float out? So from this, we saw that everything worked just like you would expect on the ground, because the surface tension of the liquid, since there's a smaller volume within the tubes, stayed adhered to the tubes. She was able to take the Zymo microbial mock community, ran a 16S PCR reaction on it, and sequenced it. And then I did the same thing on the ground. And again, we showed comparable results. So jumping off of that sample preparation, we were able to go and do the first off-Earth identification of an unknown microorganism, which is crazy to think about. It's 2017, and we haven't been able to identify unknown organisms in spaceflight until this point. So astronaut Peggy Whitson, again, took the culture plate that I talked about at the beginning with the sample collection and selected colonies off of those plates and prepared them for sequencing, did a colony PCR, and did loading on the sequencer. And then those plates were returned to the ground, where then I did our Sanger sequencing and our BITEC biochemical profiling. And the results that we got from sequencing on the ISS and the Earth matched. So we got a Staph hominis and a Staph coppedis from those samples. So this was, again, another paradigm shifting event of showing we can start using these technologies to get identifications in flight. We don't need to always return everything to the ground. But that stepping stone that we took was just looking at basing off of a culture plate. And our ultimate goal is to be moving fully away from the need to culture anything, because we know that when we're culturing things, we're biasing what we can grow off of that plate. Like I mentioned earlier, we're only using one type of agar for bacterial and fungal. We're using just room temperature. So when we know that we're limiting what we're detecting. So we wanted to dig further into a culture independent method. So going back to our ultimate swab to sequencer goal, we did this large validation of samples so that we could get more confident in our method. So 2 swabs were collected in tandem from surfaces from around the office building or in homes. One swab went into our swab to sequencing method, where you extract the DNA from the swab, clean it up, do a 16S amplification of the full 1500 gene, pool those samples and load them and sequence them. Or the other swab would go into our traditional standard operation procedure. So we plated it onto TSA, let that grow. Then we had to streak it for isolation, let that grow another day, then do an extraction of that colony. We did a 16S PCR again, but looking only at the first 500 bases, and then did a Sanger sequencing prep and loaded on the Sanger sequencer. And this is a large slide of a lot of data, but what I'm showing here is 102 different locations that we tested and doing a side-by-side comparison of what did we see in the nanopore data and what did we see in the culture data. And I wanted to highlight that we did have several locations that were negative for both culture and nanopores. That was reassuring to see that we could get negative samples. This is just at the genus level because a lot of these identifications that we get from Sanger sequencing are actually only able to identify to a genus level. The 2 main things that we find are bacilluses and staphylococcuses. Those are really common things that we find in our culture data, so it's reassuring to see that we are seeing that in our nanopore data. But to show that the nanopore data is showing additional diversity in these samples beyond what we see in our culture samples, and seeing that those diversities are things that make sense to be in an environment. They're human associated, but they might be hard to culture, things that we wouldn't normally see on our culturing samples, but are there in the environment. So with this assessment, it gave us confidence that our method is capable to do some of the risk assessments that we need to be doing with our microbial monitoring using sequencing. So moving forward, this brought us to the option to do actual swab to sequencer on the International Space Station. So this was with the biomolecule extraction and sequencing technology payload in 2018. And you can see there's a picture here of astronaut Ricky Arnold collecting a swab from this area behind the spoon that's hanging on the wall by the dining table. And so he collected that swab, he put it into the quick extract, he ran the mini PCR to extract it, again, did the cleanup, did the 16S amplification, and sequenced it all on board, all on the space station. This was the first time that we ever did that. And the data then, the other picture shows the little dots. The colored dots are the different microorganisms that we found. The different colors represent the different organisms. The size represent a relative abundance of what's there. And what we saw was so exciting because it was all human-associated oral microorganisms, things that you would really expect to see from an environment where people are eating and talking and licking their spoons and hanging them back on the wall because they don't have a dishwasher up there. They just, when they're done with their spoons, put it back on the wall. So it's cool to see that the microbes that we're detecting here match the environment that we would expect to see. So I was happy to see that it was all working as expected. 2 other samples that we collected during this test with astronaut Ricky Arnold and Serena Anand Chancellor was from an air grid and this PMM curtain that blocks between some modules. And again, from these 2 samples, what we saw was more skin-associated microorganisms, which would make sense for things that are floating around in the air and getting captured on the return grid. But the general profiles that we saw from this were similar to our culture data and data from previous ground-based assessments of samples back from the ISS. So again, showing that we can bring the laboratory to the space station and start eliminating these need for culture-based methods. So based off of all of the work that we've done and the little stepping stones of developing molecular capabilities in spaceflight, it's brought us to being able to expand from just being a research activity into a more operational microbiology monitoring setting. And this is what we're calling Biomol. The Biomol facility is owned by the Crew Healthcare Systems, or CHECS, and they own the culture-based hardware that I was talking about at the beginning of doing our microbial monitoring with cultures. So now they own a whole suite of molecular biology capable tools, from the PCR machine to the sequencer, the pipettes, the kits, and the magnetic stand. So by doing this, we are able to do a direct side-by-side comparison of the culturing method to this culture-independent molecular biology method. So with this, the crew members would collect a sample and then process the nanopore, and then we'd get the sample back on the ground and could look at it. So we were fortunate to be able to do this with 4 more crew members. And we got to work with astronaut Kate Rubins again, who did the first sequencing in space, and now she's actually getting to move on to the sample prep. So that was really special to get to do that activity again with her. We got to work with astronaut Chris Cassidy, Raj Achari, and Aki Hashido. And they really helped us expand our knowledge of how to, again, transition operations to in spaceflight, expanding our methods, and given some opportunities to do full end-to-end sample to analysis. So here's some data that we got from that testing. The picture in the top is showing the slides that we got back on the ground. So sometimes they're a little smushed because we grow them up on board and then they get taped up and shoved into the vehicle and returned to our lab here. And we processed those samples back in our lab following our Sanger sequencing. And those identifications are listed here. And then in the white box, again, those little dots are representing the different microorganisms that we found on the surfaces using the swab to sequencer method. And we saw really good similarity and matching up between the culture and the nanopore. So we found a Staphylococcus sapolyticus and some Micrococcus on this CVIS seat. We also saw that in the nanopore, along with additional things that we wouldn't be able to culture, some like streptococcuses and some Cutibacterium. I forgot to mention the CVIS seat is actually like a bicycle seat. So it's an exercise equipment that the crew member is using daily and working out a lot on. So going off of this data, it's really highlighting again that side-by-side comparison of how the culture data and the nanopore data are similar to each other, but stressing how we can use this nanopore data to expand our microbiome understanding of the ISS, and how we can use this nanopore data to complete NASA's risk assessments. And the data here that we saw with the nanopore wouldn't have altered the risk assessment that we made from our culture data. So with this Biomol, we also had this opportunity to do the first full sample to answer, getting the data analysis done on board, which is a big goal of ours moving forward, is to start implementing this more. So we collaborated with HPE, IBM, and NASA to take the data from Biomol, actually base call it with Spaceborne Computer 2, and then run our scripts and analysis. And so this is showing that we could go from full sample to answer within 24 hours, which, remember when I showed you at the beginning, our culture-based methods from sample to answer was over 30 days. So this is a huge milestone to show the massive bounds and improvements that we can be making. All of the data that I showed you previously was looking at surfaces and bacteria. We want to expand our samples, and so we've taken the swab to sequencer method and have modified it to fit water. So we can take water from the potable water dispenser, and we're going to filter it onto a 0.2 micron filter, and then you put that right into the quick extract and do the same downstream processing. And so we actually have kits that are launching in May on NG19. where we are going to be doing this water monitoring in flight for the first time this summer. And we're really excited about that, to see how does the in-flight data compare to samples that we get back, because we get this water back, but by the time we get it back, again, it's been over several weeks, and a lot of times we just see a few major microbial communities in the water system. So this will be really interesting to see, how do the water systems look. Additionally, we're flying new hardware. We're going to fly what we call MinION 2.0, but it's the MinION Mark 1C from Oxford Nanopore, again, pushing us forward to how do we build up this more capabilities in flight. And so with that, it's saying, we've building these capabilities. We have this molecular suite on the space station. What else can we do with it? So from our standpoint on microbiology, we're also expanding in ground-based research for air monitoring so that we can do the whole suite of air, surfaces, and water. We're also looking into wastewater monitoring. There's parts of the wastewater system on board that have never been assessed. So we're excited that we could use the onboard laboratory techniques to do some assessments of the wastewater. And then we've also have a lot of ground-based analysis looking at fungal profiling, and we know that that's another area we can use this for, is doing fungal sequencing on board. Finally, Biomol has another side that they are creating, is this research facility, so opening it up to other researchers to be able to use this molecular suite on board. So we have some collaborations where we're actually working at full genome analysis, some epigenomic assessments, and some shotgun metagenomics that we're planning to do on station in the next year. Moving beyond the ISS, looking at gateway habitat, the MinION sequencer is slated to be part of that gateway habitat for the microbial monitoring. It's called microbial surface monitoring. That's what the program that we're doing right now, and it's the full swab-to-sequencer methods that I showed. As part of that, we are looking at different ways that we could do increased crew autonomy, so looking at sample automated preparations. Two different projects are the SAMS project by Dr. Aubrey O'Rourke at Kennedy Space Center, and PowerBlade by Dr. Theodore Beres at the Canadian National Research Center. Those are two projects that we're really interested in. How do we reduce crew time and make a sample preparation more automated? Another big area for Gateway is looking at shelf stability of reagents. Part of the requirements for Gateway is that we have a totally shelf-stable chemistry for up to a year. So going through our swab-to-sequencing method, we were able to find a magnetic beads alternative that are shelf-stable from Agilent, and checking that out. For the DNA extraction, we're collaborating with LGC Biosearch to take their quick extract buffer and find a way to make that shelf stable. We're collaborating with New England Biolabs on their long amp TAC master mix, making that lyophilized or shelf stable. And finally, with Oxford Nanopore with their chemistries and their flow cells. So all of that is in work. We actually have preliminary studies with the flow cells at 6 months and being able to generate usable data after 6 months at room temperature, which is a very big step. And we're continuing to test that out up to a year. So there's a lot of really exciting work being done here to make this really, truly, our goal is to say anyone can use this sequencing workflow in anywhere and eliminate a lot of these constraints. Looking beyond environmental sampling, we're starting to kind of dip our toes a little bit into human diagnostics. So we were part of this experiment with Palmer Station, looking for viral reactivation in crew members, and using our sequencing method, comparing it back to the gold standard qPCR method. So this is something that we have in work right now, but exciting to just be moving a little bit beyond our comfort zone of environmental sampling. So just to close, I wanted to remind everyone that as we continue to explore, Wherever we go, our microbes will go. So microbiology is always going to be a really important aspect of spaceflight. And we're excited to continuing to keep pushing our research forward to make this possible. So acknowledgements, thank you to everyone on my team who's made this possible. We've had lots and lots of huge successes that wouldn't have been possible without everyone. Really wanted to highlight the sequencing spaceflight team. So the main people that got the sequencer on board was Dr. Sarah Wallace, Dr. Aaron Burton, and Dr. Kristen John. Especially my MinION team at NASA, all of us doing the research in the lab every day, and of course our spaceflight molecular biologists that made everything possible in spaceflight, and the ground teams. Like I said, I could go on forever and ever. But again, thank you to all of our funding sources that have made all of this possible. And thank you to all of you for tuning in today. I'm excited to get to talk to you and answer some questions. So thank you very much.
Thank you, Sarah, for that extremely interesting presentation. I hope that everyone listening enjoyed the webinar and is finding it informative. We will now move on to the last part of today's webinar, the question and answer session. Just as a reminder for those listening, please feel free to continue asking your questions, again, using the tab at the left of your screen. Simply hover over that speech bubble icon and click to ask your question. So, Sarah, the first question I have from our audience for you today is, would you suggest that this workflow could be easily adopted in a low resource or in the field settings back on Earth?
Yes, yeah, it can. That's one of our big goals at NASA is not only spaceflight, but also how can we make life on Earth easier? And so this can be fully applied in many settings. We actually have a collaboration with the Center for Disease Control. They have a lot of interest in deploying this method in hospital rooms to say, how can we do a rapid assessment of hospital rooms to make sure there's not something carrying over between patients?
Right, thank you. The next question that we have is, are you looking at sequencing for astronaut in-flight healthcare?
Yeah, so I touched on this a little bit with the work that we're starting to do with at the Palmer Station. That's our first kind of collaboration getting into looking at some crew data. It's a very sensitive area, as you'd understand, looking into crew healthcare systems and crew health data. We aren't doing that currently because we're microbiologists. That's our big area that we're focusing on. But it is definitely an area that this technology can be used in the future.
Great, thank you. The next one, sort of looking ahead, will your swab to sequencer method be employed sort of in transit to Mars, on Mars? Is that something you envision for the future?
Yeah, that's our goal. Like right now, if we don't have this swab to sequencing technology, then we don't have an ability to do microbial monitoring. We're not going to be able to send these culture plates to Mars. So that was a big driver in developing this method. And Mars is obviously a little bit further off. So our big focus right now is the moon, and we are sending this to the moon. It is on the the flight. That's what I was talking about with the microbial surface monitoring program. Like this is what we're planning to do for microbial monitoring for the moon.
Brilliant. And yeah, I'd say you touched on this during the talk, but perhaps just diving into it a bit more of, how has the sample preparation workflow been sort of configured that can be used by, team with scientific knowledge and team without?
Yep. When we were developing all of this, we have uh, everything in kind of pre-packaged tubes. So it's like one-time use. And we have these really detailed procedures that basically tell the crew, okay, collect your sample using this method, and then put your sample into tube A, and then pipette from tube A to tube B, and tube B to tube C. So it's very easy to follow protocol. It takes away kind of any doubt to the process. It's plug and play. So anyone can do this method. And we've done it with everyone from we've worked with Kate Rubens, who is a trained biologist and can do all this stuff in the lab. And then we've worked with astronauts that have never touched a pipette before, and then they come in and can handle it all.
No, brilliant. The next question just come through, asking whether any of the technology has been applied to detect foodborne organisms. So in the food that's packaged and obviously sent up as part of the launch on the International Space Station.
We haven't been using it for food detection. It can obviously be used for that for in the future if there was an outbreak, but that's not an area that we're studying.
Okay.
Mhm.
Next question, again, saying excellent presentation first and foremost. Just asking about the, how you dispose of the culture plates. So obviously that's early on, sort of prior to this method. So yeah, how did you, dispose of those? And obviously, while they were on the ISS, where were they kept and that sort of things? Any risks with obviously them being stocked?
Yes, yeah, this is something that I'm really interested in because we grow them on board. Like they collect them and they put them just in a Ziploc bag and find like a warm corner of the space station to put them in to let them grow. Then the crew have to take them out and look at them and say, hey, is there growth here? And then they typically get returned to the ground. So they get loaded into a return vehicle and sent back to our lab where then we can process them. The extra slides that don't get returned, similar thing. They just get packaged up into Ziploc bags, put into a trash bin, and launched in a vehicle that gets burned up in the atmosphere. So, no, we don't have an autoclave, but we just throw it out into space trash and it gets burned up.
So different form of autoclave, but
Yes,
same.
yeah.
So again, sort of similar concerns with culture of organisms. The next question, talking about the potential of open culture in space,
Mhm.
and obviously you mentioned that it's there isn't a fume head on the station itself, but going forward, is sort of open culture something you envision?
There are hoods on station that you can use, like a glove box. And so when we have opened culture plates on station, we've used a glove box or a glove bag to contain it because it is a very serious concern and that's one of the big passions I have about going to culture independent is that we're not growing up all these bacteria and making them more abundant. So no, I don't see us using open plates. Like we have to have levels of containment to make sure that the crew are being safe. So I think our alternative to doing that is going to this culture independent approach.
Brilliant, thank you. The next question, sort of covering obviously from the bottom of the ocean, working with some of these partners that you did to get the technology up onto the ISS. So, how important is those long-term collaborations that you have with manufacturers like LGC that you mentioned in taking this work forward to your subsequent missions?
Yeah, it's really important because we have these like really strict toxicology limitations. So when we are going through certifying things for flight, we are lucky that our vendors are open with us so that we understand what are the potential things that the crew could be being exposed to, because our life support systems are really sensitive to any kind of chemical that could be in the environment. So that's one thing that's really important to us. Another is, like I mentioned, that big area that we're getting into is shelf-stable reagents. So we are in, like, my goal is to do microbial monitoring. I'm not trying to develop a new chemical. So I like to rely on the industry to say, what do you have that's available for me? So that's really valuable to us to say, how can we use things that already exist to be applied to spaceflight or be applied to field work? So, yeah, I think those relationships are really important. All of
Great.
the work.
Great, thank you.
Yeah.
I've had a follow-up question on that, talking about sort of the shelf stable reagents. So, they ask, why is developing shelf stable DNA extraction reagents, so important for that next steps of the gateway habitat that you have in mind?
Mm-hmm. Well, so for Gateway, for ISS right now, when we send stuff up, we have freezers available on the launch vehicles or refrigerators. And when they get to station, they have the storage conditions there. So for Gateway, in the design that they're coming up with, they don't want to have a bunch of cold stowage necessities because it just takes extra power, extra engineering. So that's just a requirement for gateways to have not reliance on temperature storage.
No, thank you. The next question asks whether the microgravity environment caused any problems with your sample preparation steps.
We were really concerned about that because you see that liquids act really different in microgravity. So that was one of the very first things that we looked at is when we open tubes, what's happening to the liquids. So we're saying surface tension really helps keep the liquids in the tubes. And as long as your liquid's in the bottom of your tube, you're able to pipette accurately. And we haven't seen really any issues with it. It's been really exciting.
Thank you. The next question's again forward-looking sort of healthcare diagnostics and the potentials there. So they're asking, are UTIs frequent among astronauts, and if so, how would you sort of foresee this technology being used to detect and then potentially treat that? If so,
I don't know if there's UTIs. The crew data, like I said, is very controlled, and a lot of information. I only find out about things if a crew member like specifically will open up about it. There's some crew members that are very like sharing with things that they have experienced. So again, I think we could apply this technology to, and if there was something that a crew member specifically requested or a program, yeah, we can develop a method and apply it.
yeah, interesting. The back to sample preparation. The next question is just interested, how bead-based purification was viable in space, and were there any complications when you considered that?
That was one thing that's actually really cool for us to see is when we started doing the bead cleanups in space, the beads like bind to the magnet really quickly. We were thinking maybe it's just like something with the microgravity environment allows it to bind quicker. Just with most things, it's really comparable to what we do on the ground. We didn't see any negative impacts or anything. One thing kind of talking about microgravity prep that we made me think about is one thing we always look out for is bubble formation because, like I said, the liquids, the way they hold on to things differently, we want to try to avoid any kind of bubbles in there. And if we do get bubbles, we do a lot of like the crew flicking the tubes or like shaking them down. One crew member did like a whole arm swinging thing and she called it like a human centrifuge. So a lot of getting creative with what you have because you don't have the whole laboratory. You don't have like a little centrifuge to spin down your tubes. You don't even have like a tube rack to hold your tubes. So we use tape to like hold things onto surfaces and stuff like that.
Yeah, brilliant. So as you say, working with what you've got to get it through. I mean, you sort of touched on this with the flammability, but the next question asking, what are your specific requirements for DNA extraction reagents? Things like highly flammable reagents such as ethanol, obviously a concern. So, what is your specific considerations there?
I guess there's a whole suite of regulations of toxicology, and when we pick out a reagent, we have to send it through to the toxicology department to do a full assessment. So I don't have all of the specific information on that, but the main things that I always avoiding are alcohols or things that could be flammable or things that you would be not wanting to use in the lab, anything like everyone I know likes ethidium bromide. You don't want to smell that smell. So kind of just using your logic on things that that could be concerning. We just try to find things that are, easy to use. So that's not specific to the chemistry, but also just the application of it. that's a big area of what I'm looking for, is something that's simple to use. And then obviously something that works. That's a huge impact for us because we've tried lots of different DNA extraction methods, and we specifically for this swab to sequencer, we're putting in really low biomass samples. So we want to make sure that we're, able to process the whole swab itself. So we found that when we just like added a sample, like and mixed it around, that wasn't enough. So we liked finding something that we could put a whole swab into to be able to heat that and pull samples off that.
Oh, brilliant. Well, lots of questions coming through, but I'm afraid we do only have time for one more. And that is, have you thought about adding sort of biofilm sampling, obviously build up of these potential harmful organisms over long term? Yeah, any thoughts there as a final question?
Yeah, we're actually doing a lot of ground-based collaborations with Marshall Space Flight Center and the Montana State Biofilm Center. That's a big area that we're getting into, and our future work that we're going to be doing on board is this, we're calling it filter to sequencing. So we're getting into water sampling first with the potable water dispenser, and then getting into the wastewater systems and being able to find there could be biofilms forming in there that we aren't aware of because those areas haven't been sampled because we can't get to it on board. So this is definitely a whole new area that we're getting into.
Yeah. Brilliant. Well, I'm afraid that is all we have time for today. I'd like to thank our expert speaker, Sarah Starr-Rommel, for today's discussion presentation and this Q&A session. And a big thank you to everyone joining us online for submitting your questions. We hope that you found this a really worthwhile session. If you have any other questions, please feel free to email myself using editor@selectscience.net, and I'll follow up with your questions after the webinar. If you'd like to listen to today's webinar again, or perhaps invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.